In this video, we will discuss about microheterogeneity glycoprotein, heterogeneity glycoprotein, and non-immunogenic residues, sugar residues. Okay, so let's start with microheterogeneity glycoproteins. So, what is microheterogeneity glycoproteins? It is this is mainly depends upon the sequence of amino acid that is uh, present in the protein. You can say present in the protein, or the amino acid that is translated from RNAs. So in this macroheterogeneity is depends microheterogeneity depends upon cell lines, and cell lines and protein cell lines of protein structure, cell lines protein structure. So let us and there are other factors also on which macroheterogeneity depends. So let us consider or let us understand some examples or one examples of the cell lines. In cell lines, for example, N-glycosylation is usually occurs at NXST site. N is asparagine, S is serine, and T is threonine. And the enzyme that helps in attachment of sugar residues to this consensus sequence is OST, oligosaccharide transferase, and there are usually two kind of OSTs are present in mammalian cell lines: STT3A, STT3B. And because of presence of these two kind of enzyme, there are different kinds of macroheterogeneity. There is a possibility of macroheterogeneity in the glycoproteins. So what people have identified to reduce this macroheterogeneity is that first, if people have identified that if someone if there is a mutation in the, or someone do the mutation in the amino acid sequence to convert all the NXST site, or you can say NST, NXS site to NXT site, then the macroheterogeneity has been will be reduced significantly. And another case that people have choose is to eliminate one of these oligosaccharide transferase. Eliminate one of these oligosaccharide transferase will also helps in reducing the macroheterogeneity. And let us now discuss something about microheterogeneity. Macroheterogeneity is generally observed because of cell lines, also because of cell line and abundance of enzymes that are responsible for glycosylation. Protein conformation, residence timing, residence time in different cell organelle, you can say. So in this case, what happens is that within this single sequence of within this single general consensus present in the amino acid, there are different kind of glycans can attach, and these are mainly because of might be due to different cell lines, and this is not that much effective, or it is present, but it is not that much effect of different cell line is not that much evident. However, the abundance of enzyme present in a cell line is much more evidence, and the different competitive enzyme for the single site is the main reason why the microheterogeneity might occurs. In order to tackle this, microheterogeneity, what people have done, or what researchers have done, is that the microheterogeneity different enzymes are present. In order to tackle macroheterogeneity, what people have done is that to generate a novel enzyme, that can target specific site that can help in reduction of the microheterogeneity, However, there is no such significant progress has been done to reduce microheterogeneity on a large scale. And last is the we will discuss is non-immunogenic sugar residues. Most of the time, the therapeutic proteins that are produced is usually produced in the different cell lines, and most common of them is the mammalian cell lines, that is Chinese hamster ovary cell. In this, still there is some problem, is that this Chinese hamster ovary cell has alpha gal epitope, which can generate, or which can lead to immunogenic response in the human body. So in order to use Chinese hamster ovary cell for therapeutic for the production of therapeutics glycoproteins, we can modify this Chinese hamster ovary cell by knocking out the enzyme that is responsible for producing alpha gal epitope, that is alpha gal 1,3 galactoside transfer. Galactoside transferase. So by knocking out this enzyme, this alpha gal epitope problems can be resolved. Another problem in those mammalian cell lines is that it can produce sugar that have mu5GC, which is not usually present in the human's glycoprotein. In order to eliminate this, the enzyme that is responsible for converting mu5AC to mu5GC can also be eliminated or knocked out. Can also be knocked out. I'm not able to recall the name of the enzyme. You can get the name of the enzyme from the reference that is attached with the video. So these are the few strategies among the many strategies that are available that is been used to reduce these problems in the biotherapeutic glycoprotein production. So that's all for this video. Thank you.